This video is brought to you by Meditate AI, a medical data exchange with built-in data annotation software and services. Just remember, medical plus annotate equals meditate. Go to meditateai.com to learn more. Bacteria. These little guys are getting stronger, antibiotic resistant. It's not 1928 anymore. You can't just toss penicillin at it and hope it dies. No, bacteria these days eat that stuff for breakfast. And due to the rapid rise of antibiotic-resistant bacteria, there's been a real need for new antibiotics. But problem, it's been hard. People have exhausted all of the options. The thing about finding new antibiotics is that they found all the good ones already, so it's getting harder and harder to find new ones that work. It used to be you could find antibiotics by just plucking fungi out of the ground and seeing what's killing the bacteria. But nowadays, scientists have found that a lot of the antibiotic molecular structures are very common amongst these fungi. And so as bacteria develop resistance to these structures, it's become more and more difficult to identify novel structures that these bacteria haven't already developed a resistance to. It's becoming more and more expensive to develop new antibiotics that are just as effective as the old ones. This is a problem because it's estimated that deaths attributable to antibiotic-resistant bacteria will reach 10 million per year by 2050. So what is the medical field supposed to do here? All of the top scientists and great medical minds just can't seem to figure this problem out. Solution: starts with an A, ends with an I, it's AI. Well, not actually, but kind of. In a recent article published in Cell in February of 2020, scientists from MIT describe a deep learning approach to antibiotic discovery. And in the paper, they actually outline how they used deep learning to discover a new antibiotic. Well, if these natural molecules aren't working out, then what are we going to do? Well, scientists have turned to large synthetic chemical libraries and tried to screen those in the hopes of finding new antibiotics. Unfortunately, these libraries can have millions of molecules they've got to sort through, very expensive to curate, and are quite limited in chemical diversity. In fact, the paper says since the implementation of high throughput screening in the 1980s, no new antibiotics have been discovered by using this method. These MIT scientists actually used a neural network to identify which of the drugs in one of these massive libraries was able to produce antibacterial activity in molecules that they didn't even think could be used for antibacterial activity. They described 3 stages. First, training a deep neural network on 2,300 molecules to predict growth inhibition of E. coli. Second, they took that same model and then applied it to other chemical libraries comprising of over 107 million compounds, hoping that the model trained on these previous known inhibitors of E. coli would be able to identify new compounds that would be able to do the same thing. Finally, they ranked all the compounds that this model spit out and then set a threshold score based on factors such as prediction score, chemical structure, and availability. After doing all this, what'd they get? The C-Jun N-terminal kinase inhibitor SU3327, which they renamed to Halicin for stupid people, was found to be a potent inhibitor of E. coli while still being structurally divergent from normal antibiotics. They also discovered that it actually works by disrupting the pH gradient across the membrane of the bacteria. On a side note, the WHO has described A. baumannii, and I don't know if I'm saying that right because I'm just reading it from the paper, but A. baumannii, with 2 I's at the end, is one of the highest priority pathogens that is drug resistant, that the WHO has described as a pathogen for which antibiotics are urgently required. And what do you know? Halicin is actually a potent inhibitor of this very same bacteria. Are you smarter than me and looking to build the next deep neural network that can identify these crazy antibiotics? Well, it turns out the model they used was a directed message passing deep neural network model as described by Yang et al. 2019. This model translates a graph representation of a molecule into a continuous vector via a directed bond-based message passing approach. Don't know what that means? Well then Google it. Using artificial intelligence in drug discovery has great potential to reduce the costs and the amount of time it takes to discover new antibiotics and drugs. And this paper, and the methods described in it, reflect one of the many ways that machine learning and medicine can work together to build a greater future. This is Nguyen from the Meditate AI Laboratory, signing off. Looking to build the next great antibiotic using deep learning? Meditate AI can help. To learn more about how Meditate AI can help you find medical data or annotate your data, go to meditateai.com or subscribe to our channel to get more videos on the intersection of machine learning and medicine.